Okay, so hello everyone. Thanks for joining us for this first 2026 Assistant AI Methods for Health Data Science Seminar Series. This is organized by the Startup AI Alliance. The purpose of the group is try to aim to organize activities that empower institutions to participate more actively in AI research and leadership. Here's some like introductions of the group, and then we have the website, and then also we will upload the seminar series to this YouTube channel. And then this is the current steering committee and then the inaugural officers. Today it is my great honor to introduce our speaker, Dr. Andrew Saykin. Andy is a nationally recognized leader in Alzheimer's disease and neurodegenerative disorders research, with particular expertise in neuroimaging, genetics, and biomarker-based precision medicine. He is the Raymond C. Beeler Professor of Radiology and Imaging Science and Professor of Medical and Molecular Genetics at Indiana University School of Medicine. He is the director of the Indiana Alzheimer's Disease Research Center, the ADRD, and an IU Center for Neuroimaging. And he is also leading the genetics core of the Alzheimer's Disease Neuroimaging Initiative ACME study. So if you are doing the AD research, you must know the data, the study. And he also serves as an MPI of multiple NI sponsors consortia, including Clear AD, K-Base, and an AI for AD. And these research interests focus includes utilizing system biology approaches to understanding pathways driving AD and AD-related dementias, leveraging multimodal neuroimaging, multi-omics biomarkers, and AI-based strategies. Andy is the founding editor-in-chief of the Brain Imaging and Behavior Journal, and then he is highly cited with over 700 publications. So Andy, thank you so much for doing this for us. And then I will just some logistics before I hand it over to Andy. So please save your question to the end of the webinar because Andy has to go at 1 p.m. And then if you have further questions which do not have the chance to ask, then also please feel free to email Andy. And stop sharing, and then I can stop sharing. Okay, so Andy, it's all yours.
Let's see, is my screen coming through?
Yes.
Okay, great. Well, thank you, E, for the nice introduction, the invitation to speak to this group. I love speaking to data scientists, and hopefully I can provide some information that'll be helpful to you all. I have an ambitious title here. We'll see what we can actually accomplish today. But I'd like to just, all right, here we go. These are my disclosures and acknowledgements. Mostly just very grateful for the grant support over the past years that has enabled collection of a lot of the data that I'll refer to. I'm going to touch briefly on Alzheimer's disease. I'm sure some of you know a great deal. I know some of you do, and others may be less familiar, but interested in getting involved in analytics in the Alzheimer's and related disorders or ADRD space. The theme for our center, a lot of our work, and I think probably for many of you as well, is precision medicine. I'll touch on that concept, biomarkers, some advances in biomarkers in disease modifying therapy, talk about a systems approach, the role of AI and future directions. And all of that will be 10,000 foot, relatively brief and not a lot of data given the time. So this is an old story, not a new story. Alzheimer's disease was discovered, and reported on back in 1906, 1907. We've known about plaques and tangles or amyloid and tau protein, not since that time, but for decades now. Is illustrated here, the amyloid beta or senile plaque on the left and neurofibrillary tangle composed of phosphorylated tau protein on the right. But we still haven't cured Alzheimer's disease. There's a tremendous amount of information available. And a lot of what I'm going to do today is just point you toward databases and sources. And it'll still be scratching the surface in under an hour here. But a great source for those less familiar is the Alzheimer's Association website. als.org, which has all kinds of statistics, annual reports. There's an annual update of facts and figures. So that's extremely helpful and also gives references to many other areas. So we know a lot, and we don't know a lot, but we do know a lot about risk factors and the exposome. Uh, there's increasing work on environmental factors and other medical risk factors, exposures, substances, alcohol, sex differences, and so on. Certainly cardiovascular, cerebrovascular risk factors, genetics. So we know a lot, but still don't have perfect prediction. We also have learned a lot about preventative factors and what's protective and fosters resilience. So diet, exercise, sleep hygiene, social stimulation, and so forth, is illustrated here, are increasingly being established as protective factors. So that's all great, but we have challenges. And what better venue to talk about data analytic challenges? So, what are some of them? There's a great deal of both biological and clinical heterogeneity in dementia, and even within Alzheimer's disease as the most frequent subtype of dementia. There's also no single phenotype or marker for the disease, whether it's MRI, blood, or cerebrospinal fluid, or cognitive testing, or anything. We don't have one factor that we can just look at. So accompanying that, there's very high dimensionality of the data. The clinical data is high dimensional, multiple factors. The biological data certainly is, and I'll say more about that. And the imaging data, brain imaging with MRI modalities, PET scans, and other tools, also are very high dimensional. The big challenge, and actually what keeps me up at night scientifically, is in all the sea of data and information, the explosion of big data, where are the causal factors? What's really driving the disease? And I'll come back to that theme. Some of the other challenges are that it's a challenge and an opportunity, really. There's a very long preclinical phase of the disease in prodrome, and that's a potential diagnostic and therapeutic opportunity. Yet we have to figure out how to optimize that. There is extensive longitudinal data, which comes with its own challenges. And then there are the things that many of you who are biostatisticians, really know better than I about data sparsity and missingness and the issues of less represented populations and biases, collinearities and nonlinearities, and that's scratching the surface, but just to name a few of the challenges. So I've been trying to figure out how to capture all this complexity, and I finally realized it's a giant hairball. So this kind of illustrates my view of the problem. Everything at some level is correlated with everything else. And there's so many different layers, from social networks to clinical data to genes to proteins, and on and on and on. All these layers, electronic health records, and you name it. It's very complex, and we have to figure out. I mean, you may have a better analogy. Mine is we have to figure out cutting planes through this hairball to start to dissect it in some way. You have to hold something constant, I think, to get a handle on it. But we do have this problem of a lot of heterogeneity and complexity of these interrelated pathways. So we have a review that's currently just being revised for one of the journals that looks at all of the literature over the past 15 years. And essentially, there are a lot of biological pathways in the literature that have been seriously investigated. Two big slices of the pie are amyloid and tau, plaques and tangles, but there are a lot of other slices. Immune dysregulation and neurodegenerative processes, oxidative stress, vascular, certainly, other genetic risks, diabetes, and I could go on and on. But you can see the list here and glance at it, and they'll look familiar. There are a lot of different ways of slicing and dicing these biological pathways, but clearly this is complex disease. So the goal, the overarching goal, I think, for many of us is really precision medicine. And going from the hairball to precision is the grand challenge. So we want to take that heterogeneity, go from a one size fits all, take this pill or injection or infusion strategy to a strategy that really fine tunes and tailors the therapeutic approach from whether it's medications, lifestyle modification, other factors. It's what gets the right treatment for the right patient at the right time, as precision medicine is usually summarized. To do that, biomarkers are essential. And there are a number of different types of biomarkers. So there are predispositional biomarkers, our genetics that we're born with, genes like ApoE, the overwhelmingly important effect size in sporadic Alzheimer's disease. But there is also the rest of the genome. Um, then there are diagnostic biomarkers like PET scans to measure amyloid and tau in the brain, cerebrospinal fluid, and now blood-based biomarkers. So those give an indication not only of your genetic susceptibility, but where you are in the trajectory of disease if you're going down that pathway. Then prognostic and predictive biomarkers are really important when we have treatments to try to understand somebody going to have a very stable, slow course, they're low risk for progression, or is it going to be one of these people that they come in for a diagnosis and within a year or two they kind of crash and burn and come to the neuropathology core of our center, and we see very slow and also very rapid progression. So understanding what's driving that is really important, and then we want to ultimately find out uh, what what profile from all these factors would predict the optimal response and minimize adverse effects for particular treatment strategy. So these are all familiar concepts. You could apply the same ideas in oncology, where a lot of this work has been done, and to other disease areas. But that's the goal. And the whole field of Alzheimer's disease has really moved to a biomarker-based framework. There are several important position papers um, by Cliff Jack and colleagues, and it was the first one was proposed in 2018. It was a bit controversial to go from clinical testing with memory and other measures and history, make the diagnosis on that basis, and move to use of biomarkers. But when you think about it, there were hundreds and hundreds of failed clinical trials, and a lot of the reason for those failures were probably that people didn't actually have the target pathology. Usually it was amyloid as an anti-amyloid therapy strategy. So if 20% or more didn't have amyloid in their brain and that was the treatment, that's going to really attenuate the effects and make it very hard to detect a positive trial result. So now we have a framework where people have a biomarker-based diagnosis or confirmation of diagnosis, and there's a set of primary core one and two, and then some additional non-specific biomarkers and co-pathology biomarkers. So amyloid and tau, A and T, are really the whole, by definition, the hallmark of Alzheimer's disease. So those biomarkers, take precedence. However, um, neurodegeneration, or N, and inflammation, immune dysregulation are also increasingly recognized as important, as is vascular pathology. And just very recently, because there are new CSF and skin biopsy based techniques for looking at alpha synuclein, now synucleinopathies are another area that are being coded, an S factor. So the alphabet keeps growing. We have more biomarkers, better biomarkers, and that's really going to help in terms of developing precision medicine. But all of this is generating tremendous volumes of data. And this is, again, where data science is incredibly important in terms of optimizing the yield from all of this very rich data. So for ATN and V, where we've been for a while before adding to the alphabet, amyloid, tau, neurodegeneration, and vascular, we have imaging biomarkers. You can see a amyloid PET scan on the left and a tau PET scan on the right. The reddish areas reflecting the deposition of amyloid and and tau pathology in this symptomatic individual. It's one of the first people we studied with amyloid and tau PET here at Indiana University. And then atrophy can be measured very specifically to different brain regions. And then in the lower right, you see the pretty extensive white matter pathology in this individual that all reflects cerebrovascular involvement. And there's a lot of interesting issues about whether it's additive, just more pathology of each type, is bad for your cognition, which I'm sure it is, but are there also interactive synergistic effects? And that gets trickier to ascertain. So an important principle is that there's evidence of biomarker changes in Alzheimer's disease decades before a person becomes demented, and even before they become symptomatic. So some of the early modeling of this was done in autosomal dominant Alzheimer's disease, which is only about 1, maybe 2% of Alzheimer's disease. But because of the familial nature and the ability to predict when an individual with a mutation, deterministic mutation, will develop the disease and become symptomatic, it allows for modeling. The same thing's been done as well in sporadic Alzheimer's disease later. But essentially, these biomarker curves for amyloid and tau and other measures start to show changes about 20 years before a person develops symptoms. So that's a really important opportunity for early detection and early intervention. Some of the major developments, I mean, I used to give lectures and talk about a blood test for Alzheimer's disease being the holy grail. I was doing this probably back, at Dartmouth, where I was 20 years ago. And now it's not just a dream. We actually have FDA cleared blood tests for Alzheimer's disease. They're not perfect, but they keep getting better, and there's rapid progress in this area. So there's phosphorylated tau 217 in blood developed by Jeff Daig when he was at Eli Lilly. He's now in our center here at IU. Was really a landmark in terms of detection in blood. And there are other blood-based biomarkers as well. They can be used in combination. But this is really a new era in having something that doesn't require a lumbar puncture or PET scan in nuclear medicine or neurology and so on, but could be deployed in primary care for early screening. So there's a lot of work in that area. The other huge development in the last few years is finally the successful clinical trials for disease-modifying therapy, in this case, anti-amyloid therapy, where there are two approved and marketed drugs, lecanemab or Leqembi, first in 2023, and then that was followed by donanemab or Kinsula in 2024. So these drugs are successful in slowing progression in symptomatic individuals with mild cognitive impairment or early stage dementia. They don't cure the disease, they don't flatten the curve, but as you can see in the left, the lecanemab versus placebo, it does start to flatten the curve, at least slow it down. So then the question is, what else can we do to understand the disease and intervene, perhaps with combination therapy, lifestyle factors, other strategies that would really flatten the curve? And I think, as I alluded to earlier, understanding what actually is driving the amyloid and tau plaques and tangles in the first place, and the cognitive decline, is going to get us to the eventual prevention strategy that we need. But those are exciting developments. How do we figure out what are the drivers? Well, I think we have to, the field has been actually criticized a fair amount for focusing like a laser on amyloid and secondarily on tau. I don't see that as a problem. I think that's ultimately proven successful and very informative, but we need to understand what's driving those changes to begin with. And we have to also look at the bigger picture. So I would argue that a systems view, multiple organ systems, looking at the periphery, the entire body is also really important in understanding what's going on in the brain. These are not isolated compartments, but we have to get the big picture at a multi-organ level, multi-systems level, and even different cell types and different brain regions, and what's happening in different organs. Um, I like to say, as a neuropsychologist by training, I never thought I'd be collecting stool samples to understand Alzheimer's disease or other brain disorders. But we're in the era of looking at the microbiome. There is a gut-liver-brain axis. We're collaborating with the hepatologists who are the experts in liver pathology now. So we have to think out of the box and look at all these different components. So. Um, the advent of high throughput assays for various omics is really opening the door. I mean, so many cohorts now are undergoing whole genome sequencing. We certainly had a productive era of genome-wide association studies, or GWAS, and now we're filling in for rare variants with within the 3 billion bases of the human genome. Um, but beyond that, we're now in the era of functional genomics, and there's a lot of intense work going on looking at the transcriptome. How are these genes expressed and under different conditions? Both in the, I'll say a little more about this later, but not only in the brain, but also in the periphery. So we are in one project called Clear AD, trying to link the brain transcriptome in multiple brain regions at a cell-specific level with what we can read out in blood using PAX-Gene2 based assays for the transcriptome in blood. So I think there's a lot of opportunity to connect the central and peripheral statuses that have yet to be exploited. And my hypothesis is that it's not going to be one-to-one. In some rare cases, we may see the same protein in brain or in tissue from brain that gets to the periphery through extracellular vesicle or some other method. But I think a lot of what we're going to see is connectivity between what's happening with multiple omics in the brain and in the periphery, but it's not going to be one-to-one, and we're going to have to figure out the translation there. And maybe new AI strategies are going to help us with that. That would be certainly something hopeful to take two very, very complex sets of vectors and understand the relationship between them and how one might be predicted from the other. Well, we'll check back. Some of you are probably working on this. We aspire to this, and maybe we'll have some answers over the next few years. Anyway, there are other layers. There's also proteins, and there's been a lot of growth in high throughput proteomics as well to understand the protein products of this fundamental biology. And then epigenetics, lots of assays, both in brain, and all these things are being done in brain and in blood, and sometimes in other tissues, CSF, depending what it is, or elsewhere, skin biopsies, and so on. And then metabolomics and lipidomics are proving really informative and important, and are closely tied to the gut microbiome as well. And I always like to, as being in radiology is my primary and leading an imaging center, I always like to put the connectome in there as another important omics, right? Because the actual substrate of all the cognition that we're concerned about and how it's impacted by aging and pathology is basically the brain as a computer with structural and functional connectivity supporting that processing. So lots of data opportunities here, right? And one of the things that Dr. Zhao asked me to do is to talk about data resources, what's out there. So I put some slides together, basically just it'll be kind of superficial, but just pointing you, and you'll have the URLs for all these things that for anyone that isn't familiar, but there's a lot out there. GAIN is a great place to start. I'll just show you the websites for these. The Alzheimer's Disease Neuroimaging Initiative, the Alzheimer's Disease Coordinating Center that links together all the Alzheimer's disease research centers or ADRCs like ours around the country. Um, the many large-scale genetics and omics systems biology related initiatives. The NIA has been great in supporting a lot of this work over the last decade. And then also now, the Alzheimer's Data Initiative, ADDI, and Gates Ventures has been a big sponsor. There's a lot of private sources that are now funding some of this work, and many other national and international programs. So this is going to be selective, and I'm mostly going to talk about human data. There's a great wealth of preclinical data from animal models, increasingly stem cells, and all sorts of other things. But just to focus more on the clinical side for today. So let's talk about gain. This is the kind of splash page for GAIN. The website is posted right up there at the top. And you can kind of surf over this, and it's basically a tour of all these different cohorts. Now, you can't just click and download the data from all of them. There's data use agreements and various things that you have to do. But this is sort of a one-stop shop that gives you a big picture of all these data sets and databases. And those who may not be familiar, I would highly encourage looking at this. If you're just thinking about design of a study, this would be a great place to start to kind of see what's out there. So here's an example. If you were to click on ADNI, the Alzheimer's Disease Neuroimaging Initiative, see it'll change the map from the global map to where the ADNI sites and where some of the cores and coordinating facilities are. And it'll do the same thing for all these other cohorts from around the world. So it's really a great resource. So ADNI, we celebrated our 20th anniversary a couple of years ago. It's been a really productive study, and I started as the site PI at Dartmouth Medical School just before moving to Indiana, launching a cohort there. So there's been about 60 active sites. They've changed a little bit over time. And now there's several thousand individuals enrolled. There have been 5 funding phases of the Alzheimer's Disease Neuroimaging Initiative, or ADNI. I think it's a pretty well-known study. Many of you are probably familiar, but it really led to these sort of classic biomarker curves generating the longitudinal data from CSF. Also, there's annual blood tests and then annual imaging studies, which really led to looking at a lot of these different biomarkers. They've evolved over time. The sampling has evolved. The technologies have evolved. We only had amyloid PET using a Pittsburgh compound B on a portion of the sample in the first part of ADNI. Later, everybody got amyloid PET with an F18 tracer. Then in the third phase of ADNI, we had tau PET. So things have changed over time. In some cases, because the biospecimens are banked, the biomarker core that has the fluid biomarkers is at Penn. The genetics are here at Indiana. But it allows us to go back and interrogate these samples, and this happens all the time. They're in the freezer to yield new insights. A big criticism of ADNI was that it was kind of homogeneous. It's largely people of European ancestry who are more affluent, come to academic healthcare centers and clinical trial sites, and so on. It was thought there wasn't, representative vascular pathology, although the truth is, if you look, there's plenty of it that can be measured with especially with white matter hyperintensities on MRI, but also with other measures. Uh, but by ADNI 4, we've really tried to focus on increasing diversity. So 50% of the individuals enrolled in the current phase are now from underrepresented groups, largely African American and Latino American samples, but also some other groups. So that's a really important development because biomarkers may not work the same in all populations. whether based on genetic ancestry, environmental factors, or other things that we don't understand yet. So it's really important to understand biomarkers and ultimately for trials to understand how different drugs behave in different populations because one size probably is not going to fit all, and we need to understand that. So you can go to the ADNI website. It's ADNI. We're all very proud. It's been the forefront of open science. As you probably know, we made all the data available without an embargo, just after initial QC. And it's led to, I think, something like 7,000 papers by now. It's just been incredibly productive that way from the whole scientific community jumping into the data and really going after different questions. So it's still there and growing. And we're planning ADNI 5 at this point as we speak. So one can go to the ADNI website, and I've got the URLs on all these slides. So for anybody who might not be familiar, you can find the data use agreement, and which is very simple online form, and then the data characteristics and put in a request for data. Um, so here's just a few examples of the current website, showing the ADNI sites, the current 60 and where they are. There's for researchers, a place where you can go to learn more about acquiring data or even biospecimens. Um, all of the different modalities shown in the lower left here, including the core that I lead, the genetics core. All that information is on the website. There's a lot of data. It's pretty complex and it's evolved over 20 years. So we're constantly trying to improve the description of all this information. I'll admit it's not perfect, but we're trying to make it more and more user friendly. And Arthur Toga, who leads the informatics core, has been working hard to try to develop systems where you can go in and query because things may be spread out in 50 different spreadsheets, but this gives you a way to link things up without having to do all of that manually. So I might be biased, but it's a great resource in my view. Um, the National Alzheimer's Coordinating Center, or NAC, just having its 25th anniversary, uh, is a great source of information. That's where all of the data from the about 36, 37 ADRCs that are currently funded around the US upload their data. And for a while, it was fairly primitive, to be honest, but it's now, in the last 5 years, it's gotten really sophisticated. I think it's still a bit of a work in progress, but they've redone the entire platform and are linking it up to other databases to make it easier to correlate the cognitive and clinical data, which NAC has always had, with imaging, with structural genetics and functional genomics and other systems level data that are spread out in different National Institute on Aging, NIA funded databases. So there's a lot of connectivity, but just some of the stats from the splash page, over 54,000 participants since the inception, right? Over 200,000 clinical visits, uh, over 8,000 neuropathology exams. So it's really quite an extraordinary resource and getting better and better. Uh, this is the aspiration to really make it the proverbial one-stop shop for ADRC data through a so-called data front door, connectivity to information that comes from all the different cores from the centers. Ultimately, it's not there yet, but to integrate we have a pilot program to look at integration with electronic health records. There's a pilot on digital biomarkers and new assays coming online as well from blood. So the idea is that all of this will be integrated and link up to portals that have, the imaging information, biospecimens, and affiliated studies. It's not all there, but this kind of presents the vision, and a lot of it's working. It's really come a long way. As one example is an imager that, very interested. No, originally, none of the Alzheimer's centers, had standardized imaging. Everybody did their own thing. But ADNI spent years working out standardization of imaging for clinical trials in Alzheimer's disease. Eventually, the centers adopted this, and the NIA funded something called SCAN that basically is standardizing the standardized centralized Alzheimer's and related dementias neuroimaging, or SCAN, that standardizes the MRI and PET scan data. So now there's, I think just looking in the lower left, over 7,000 participants have data in this new framework. There's over 6,000 MRI series, and over 3,000, almost 4,000 PET scans. So there's a lot of data. It's starting to accrue rapidly now, and it's standardized, and also it's partly harmonized by SCAN, but it's further harmonized as well by something with a long name, the Alzheimer's Disease Sequencing Project Phenotype Harmonization Consortium. It's a mouthful, or PHC, which is working on harmonizing all of the data from the different NIA-related cohorts, everything from uh, cognitive data from different neuropsychological test batteries to clinical measures, vascular assessments, to MRI and PET and fluid biomarkers, putting all of that together. So that's a huge transformation as well. Um, Clarity is relatively new. This is like a massive R01 type science initiative that's piggybacked on the ADRCs, and is paying for uh, standardized assays at each of the ADRCs. So people enrolled in Clarity, which are a subset of each ADRC, then undergo amyloid and tau PET, 3T MRI with a specialized set of sequences, blood biomarkers, and more, so that one can get a deeper understanding of the heterogeneity of disease. And eventually, the goal would be as well to follow people all the way to brain donation and incorporate that as well. So these are the goals of Clarity, which is relatively recent. I think just maybe in its third year now. So data is still being collected, but will be posted to all the NAC related websites, SCAN, and so on. There's been huge progress in genetics in the past few decades, starting with the discovery of ApoE and the autosomal dominant genes in the 90s. If you fast forward, through the GWAS era, from gene chips and arrays to functional genomics, and then to really large-scale sequencing initiatives, now there's been incredible growth. The Alzheimer's Disease Sequencing Project is working on a white paper kind of summarizing this whole history now that hopefully will appear in the next few months. So this progress also allows, I think somebody was unmuted. Anyway, I won't go through the specifics of this, but you can see all of the progress and all the data from the Alzheimer's Disease Genetics Consortium that was kind of the first phase, and then now the Alzheimer's Disease Sequencing Project, and then the functional genomics and also machine learning consortia affiliated with the ADSP, or Alzheimer's Disease Sequencing Project. All of that is available on NIA websites and is growing very rapidly, including that harmonized data from the PHC. So where can you find some of these things? The NIA designated site for genomic data is called NIAGADS, or NIA Genetics Alzheimer's Data Storage Site. Lisan Wang at Penn directs that resource. And this is just another timeline showing releases of data. Um, but it's growing at an extraordinary pace. Over 50,000 whole genomes now linked to a lot of detailed phenotypes are available. So there's some work to do with getting access to the genomic data, but there's a clear pathway in place to obtain those data with appropriate safeguards and so on. And it can now much more easily be linked to the clinical phenotypes at NAC. And both all of ADNI is a member of the ADSP and has the data at NIGADS. All of the ADRCs contribute data to NIGADS and many other NIA-funded studies as well. So a great resource. The website is there at the bottom. Uh, Functional genomics tends to be on the Sage BioNetwork's AD Knowledge Portal. Um, and that's another important one. It's not quite as simple as structural genomics being at NIGADS and functional genomics and multi-omics being at the knowledge portal, but it's somewhat like that. And I think that's generally the vision for this. Then it becomes important to be able to sorry for the noise here to be able to link all of this data together. And I think the tools are becoming better for doing that. There's still more work to do. If you want to look at structural genomes and relate them and rare variants and relate them to functional genomics and imaging data and cognition in the same person, those are some of the challenges, but they're worthy ones and they can pay off. There's also a lot of systems biology data from animal models, from model AD and mice, Marmo AD, a marmoset model. There's new stem cell-based initiatives, and drug development, medicinal chemistry related initiatives. All of this NIA supported and also in part sometimes with partnership with pharma and biotech in the AMP-AD area or Accelerating Medications Partnership. That's up in the upper left here. But these are active links on the Knowledge Portal website. So I encourage you to explore them. The program I alluded to earlier, linking transcriptomes in brain and blood, Clear AD, which I'm one of the MPIs on, that one we are gearing up over the next year. We will be uploading transcriptomic data to that site as well on the Knowledge Portal. So a few other resources. Another major one from the private sector as a partnership is the Alzheimer's Disease Data Initiative, or ADDI. And that is growing rapidly. There's recent capture. There were 310 data sets, so almost 6 million participants represented there. And there's an enclave with providing computational capabilities. So I would encourage exploration of that data. One of the new data sets that's represented on the ADDI platform is called the Global Neurodegeneration Proteomics Consortium, or GNPC. And this was sponsored by Gates Ventures and J&J Pharmaceuticals initially. Now there's more partners. It's going to a second phase. But what they did was essentially collect standardized proteomics, high throughput proteomics data with the Soma Scan 7000 analyte panel on about 35,000 individuals, with mostly from Alzheimer's related studies, but other aging and neurodegenerative diseases as well. So it's an amazing resource that we're very privileged to be part of from Indiana. ADNI is going to join that for the next phase. Our K-based consortium, that's an ADNI-like study in Seoul, South Korea, is being profiled right now. And the next phase is moving to for any of you proteomics mavens, it's going to have the Soma Scan 11,000 panel platform Olink 5000 protein panel, the Alamar very high sensitivity panels with neurodegenerative and inflammatory markers, all on the same from the same plasma sample, same visit for many of these cohorts. So it's really exciting. It's the largest proteomics database by far available in neurodegenerative diseases. And there was an embargo while it was being put together. The first overarching papers came out, but that was 1 year, and the data was released last summer. So this is available. You can go there, explore, and obtain those data, or well, you can compute on it within within the enclave. So I mentioned Clear AD. You know, we're trying to address the iceberg problem here to dissect the complexity. And as I mentioned, really look for drivers. And I think, you know, we have a lot of statisticians, I'm sure, listening to this talk. So, you know, it's as one statistician friend said years ago at a site visit, it's customary to have hypotheses in science. You know, we're in the era of high throughput discovery and exploration, but they're not mutually exclusive. So what we tried to do in Clear AD is to have hypothesized pathways and prioritize them. It's not like we're not going to turn over other stones, but basically we had sort of a inner core of what we thought were most important for immune dysregulation, synaptic integrity, cerebrovascular biology, and white matter myelination. We said those are our top four. And then all the other ones, you know, neural transmissions, oxidative stress, epigenetic factors, DNA repair, all the other hypotheses out there from that pie chart I showed you earlier, viruses, infectious, you know, you can go on, autophagy. But all of those are the second tier. And then, you know, unbiased discovery would be the third tier. So we tried to be interested in your thoughts, you know, and if not today, I'd be happy to hear from anyone that has thoughts about this as a way to structure, put some constraints, yet not be so limiting that you can't discover something new. I think that's one of the big challenges. And we're then applying this framework now to transcriptomic and other omics, multi-omics data from both brain and blood. So the goal again is like, you know, everything is going to be correlated. We know that in some ways. It's the exception when there's no relationship probably, but what are the drivers and what are the passengers? You know, that's the big question because you want to find whether it's by hub gene or some other mechanism, you know, what are the causal patterns here among all of this biological complexity? So don't, I'm not going to give you the answer to that, but it'll be on the quiz. I think, seriously, this is like the grand challenge for all of us as a field, right? To figure out what is really driving all of these different complex processes. And it may not be the same in everybody. And that might be also a factor related to genetic ancestry, to environment, the exposome, and other factors. But we should try. Uh, I think there's great promise in applications of deep learning, machine learning, other AI strategies to this. Been privileged to be one of the co-leads of a consortium that's got a long name in the upper right. The cute short name is AI for AD. That was funded as part of the ADSP, and we're waiting to hear about a renewal. But is this multi-PI, multi-institutional strategy to try to really go after some of these problems from understanding the phenotypes, from imaging, from cognition, genomes, and so on. So here at Indiana, in our group, Tejo Joe has led uh, our uh, our efforts in applying deep learning. Just a couple of quick examples that are, you know, published. So one is applying, you know, deep learning works really well for images, and it's certainly had a huge impact in radiology in general. But we've, you know, tried to look at tau PET scans in Tejo Joe's work to see if we could dissect informative features that might be useful for classification. and that was successful. And it also fit um, known factors because on the right you see conventional statistical parametric map of tau PET. You see the medial temporal region showing up very strongly in patients versus controls. Over here, this is this is a feature map to try to do an informative AI. And it basically pulls out the entorhinal cortex region very nicely as the primary site using multi-layered 3D CNN strategy. So just these are really just examples. Another is, and maybe probably a tougher one is the genome is basically a vector of 3 billion bases. It's really tough, but we've been trying different strategies, again, led by Dr. Joe, to try to figure out how we can interrogate the genome to discover patterns that may not be picked up as readily by conventional, massive, parallel, classical statistical genetics. So this one is one example. It's a sliding window association test that kind of passes through the genome using CNN deep learning strategy to try to pick up features. And Tejo Joe has several other new strategies as well that we're trying. So, and many others are working on this. I think this is another one of these grand challenges to how do you really extract the important features out of these incredibly long vectors that have interesting patterns of linkage disequilibrium or spatial autocorrelation within. So that's really interesting. And I think this is going to be fruitful in the coming years. Then ultimately, this is a busy slide, but essentially we have to boil down all that complexity into a few numbers in order for it to be meaningful. So like to take the 20,000 genes in the genome, well, we can't tell somebody, well, these 6,000 genes are good, these 5,000 are bad. What we have to do is boil it down to a risk score. So a polygenic risk score that reflects person's susceptibility. It would be something that could be explained to a patient in an exam room and be informative, where anything more complex than that will be tough. But we can do this it's been done with polygenic risk scores, but it could also be done with transcriptome risk scores, biological aging clock scores, and other factors. And I think this is the future where there'll be a profile. It can't be too complicated, but basically a profile of key features that we could explain to a patient and would be helpful for shared decision making. So I'm wrapping up here, and all of this then has to come together where we look at different patients and understand their profile to target their treatment, both biological treatments and lifestyle interventions and everything that we can possibly offer. But we need to integrate all that information from molecular profiling, lifestyle, clinical stages, digital metrics, which I haven't really talked about, but are of course exploding, for mobile apps and computer-based assessments and tracking and so on. But all of this information has to converge in a meaningful way to profile an individual and then help guide their treatment. So I think that's where we're going. And there are a lot of challenges. I got my hairball up there on the right. But I think a lot of the classic strategies often play a great role, clustering and dimension reduction. And then AI and ML modeling, I think, are going to, open up new vistas. Um, we need to optimize our selection of these markers. We can't do what ADNI does and try to get everything on everybody. That's not possible. We need to find the most cost-effective, efficient strategies for the future. Data dimensionality is a very well-known problem to this audience. Um, we need strategies for the longitudinal data, which may be sparse in various ways. And given the very long preclinical prodromal phase, also, it may show up in electronic health records. This is more of a real world data problem than it is what we would see in a clinical trial. unless we know somebody's at high risk and are following them. And then, there's all these other challenges that are well known, I think, to this audience about data properties, biases, sparsity, missingness, and so on. And I think this is where data scientists really can play a huge role. I think in the end, to understand these causal factors that I've been talking about, we need replication to show that it's real and reproducible, and then validation, which doesn't, to me, you may feel differently. I don't, the leave one out thing is okay as a first step, but ultimately you want to see validation in independent cohorts, independent data sets. When something keeps showing up in multiple contexts, right, then it becomes more believable. And so in many cases, then you ultimately need molecular validation where somebody has to do the tough slog of doing an experiment and actually making sure that some molecular mechanism is behaving the way we think it is, or refuting that. Uh, anyway, the goal eventually is to pull all this together for medically actionable signals that we can use in precision medicine. And I think the way we get there is a collaboration of data scientists and clinicians, clinical researchers. So that's one of the reasons I was excited to talk to this group, because I think that's the way forward. Nobody can do this by themselves. And I want to thank all my colleagues from all these consortia that we have the great privilege to participate in for the data generation, the ideas, the analyses, and so on. It really does take a community, and through collaboration, we will get there and make a lot of progress. So I think I've run over a bit. I want to thank you. I'll stop sharing here, but Dr. Xiao will make my slides available, and I'd be happy to hear from you if you have some thoughts. If you think I'm off target, if you have some ideas for the problems and challenges I throw out there, we would welcome interacting with you.
Thank you so much, Andy. This is great. I think we still have a couple of minutes for one or two quick questions.
Sure.
I think there are two questions in the chat. Maybe Jacob, do you want to unmute yourself and then ask the question?
Sure, yes. Thank you for the talk. Can you hear me?
Yes.
Yes.
Yeah, so I really appreciate that. And I said go Hoosiers, so I hope we prevail tonight.
Thank you.
Yeah, so really appreciate this in terms of where to go to for data, but very interested, especially in the stats talk. What is an example of methods driving scientific insights, as opposed to, I'm trained as a classic statistician, but I'm actually in AI research right now, and it's very hard to traverse. And so I'm at the gym, but traverse different, I guess, cohorts of people. And so how do you foresee the future of methods driving scientific innovation? Thank you.
Yeah, well, it's a great question, and it comes up a lot. I mean, we've got all these powerful tools, but then I think you're asking, and I ask the same question, like, what are we really learning? And many times I've told people, a little regression model that's been around for forever might be suited to this purpose, and you don't necessarily need a seven-layer neural network approach to something. Um, I think matching the methodology to the problem is really the key. And I don't think we know yet the full power of AI strategies. So I think we're going to find out the answer to your question. I don't have the answer now, but it's really the right one. And I think in general what we have to do is match the methodology to the problem, stating sort of something that's obvious.
Yeah, I have a questions, Andy. How are you?
Sure. Thank you.
Yeah, I think it is great. I think AI really push the whole and research paradigm is very fast, and also in terms of both the depth and also the width. And one quite question I observed is when you have the data, in particular the data from different studies, it's very difficult to do straight, just combine the data together. There's a lot of heterogeneity inside. And some, the data is not from the generalization purposes, there's sometimes when you're looking at a small portion of the populations, you could observe pretty elective results. That's way confounding many things. It's not about, I find that the statistical or epidemiology approach is more important at the beginning when we design and also try to do integration instead of using AI tools or other tools. Tools itself is a secondary. If you at the beginning the bias is already over there, I don't feel that the scientific conclusion from this bias samples is important, or you need the right conclusion for the final discovery. I think the people in the field didn't emphasize this that much.
I appreciate the comment. Yeah, I agree. Well, I have to go introduce the speaker, so I apologize for not being able to stay longer. Please feel free to get in touch, and I appreciate the opportunity to speak with this group.
Yeah, so Andy, so please feel free to leave. So so we are so sorry. So Andy has to go, and then please feel free to email Andy if you have any question. For the rest, so can you just stay for a minute? They'll just do a quick preview of the next seminar. So our next seminar will be on February the 9th. So we will have Dr. Robert Cass to give us a webinar. We will move from the macro level of like a human neuroimaging data to the macro level and neuronal level data. And then just please join us next time as well. And then thank you so much, and then thank you all. Great. Bye, everyone.
Bye. Thank you.